Hi. One of the most fundamental open biological questions is what determines the organization of genes in eukaryotic genomes. It has been shown that the distribution of genes in genomes is not random. However, until recently, the most studied form of genomic organization was the linear organization of genes along chromosomes, adjacencies between genes, and clusters of genes. And today I'd like to talk about gene organization in 3D, its relation to gene expression and gene function, and their evolution by studying whole genome chromosome conformations. So recently we gained the technology to study genomic organization at unprecedented resolution thanks to chromosome conformation capture and its high throughput variants such as HiC, for example, that were developed in Job Decker's lab. Briefly, this technology uses next generation sequencing in order to measure the frequency or the probability of observing a physical contact between two DNA fragments inside the cell. And we utilize these data in order to study the relation between function and expression and genomic organization in my work. First, I'd like to talk about the significance of the relation between gene organization and gene function. There has been very exciting evidence recently for gene regulation in 3D, and the extent to which global genomic organization is important is still being explored and studied. Recently, for example, we proposed a novel metric for functional similarity between genes. We called it the codon usage frequency similarity, or CUFS for short, which measures similarity in terms of expression patterns and gene function by comparing codon usage and amino acid usage in sequenced genes. And we showed by utilizing HiC data from 5 different eukaryotes, from Anthony Blau's and William Noble's paper, from Kenichi Noma's papers, and from Job Decker's lab, and showed that indeed this metric is very highly correlated with 3D genomic distances between genes. It is, of course, also highly correlated with annotations of functions and gene expression. This is why we used it. And you can see here examples of the strong correlations and strong relations that we get by looking at functional distances on the x-axis and distances that were obtained from HiC data sets. This result emphasizes the importance of global genomic organization and shows that this a very strong level of organization that exceeds what has been reported so far. We also studied, our hypothesis behind this result is that similarly expressed genes are colocalized in 3D. They have similar functions, similar transcription levels, and similar translation levels, and they co-evolve. And over the years, they develop similar patterns of codon and amino acid usage. And so when we come to analyze HiC data and measure CUFS, we see a strong relation between the two. In addition, we analyzed pairs of organisms in order to understand better the evolution of function and organization. We used 2 fungi and 2 mammals, as seen here in the figure. When we look at the correlation between functional distance and 3D distances in human, for example, we see a strong relation, as seen in the previous slides. However, when we switch the functional distances to that of the orthologs in mouse, we see that this relation becomes much weaker. It is still significant. We see here 3D distances in human versus functional distances in mouse, which comes to show that the two don't fit as well, and that there is some probably coevolution between function of genes and organization of genes. In addition, we showed that CUFS distances are very strongly conserved between this pair of organisms. And finally, we showed some level of conservation of genomic organization between these 2 organisms. taking these two results together, we wanted to apply them to the problem of 3D genome reconstruction, which is one cool way of interpreting high-C data and studying 3D organization. This particular method of reconstruction has been proposed and beautifully applied a few years ago in William Noble's lab. Briefly, in order to reconstruct a genome, one converts high-C distances to nanometric distances and poses these as constraints on chromosomes comprised of beads on a string, as seen here in the zoom panel. And then a nonlinear optimization problem is solved in order to find a model that minimizes the error with respect to the input distances. Our suggestion here, our novel approach that we suggested, is to try to predict distances in 3D based on the two results that I just showed you. One is the conservation of organization between organisms, and to this end we took S-Pombe distances and tried to project them over to the S. cerevisiae genome, and then reconstruct the genome uses these distances. And the other one is using a functional similarity, an expression similarity between genes as measured by CAFs. We wanted to check whether our integrated models that contain both high-C data and additional data that was predicted indeed are more accurate than baseline high-C models. And to this end, we used 15 previously published results on S. cerevisiae genomic organization. And indeed, on most of these tests, we see an improvement, a stronger signal for integrated models. For example, you see here what we get for a high-C only model in green, some colocalization of early replication origins. And when we add predicted distances, we see a stronger signal, significantly stronger, of colocalization of early replication origins. And the same goes for many other interesting functional results. So up to now, I tried to take advantage of the conservation of genomic organization between organisms. And here we want to study changes in the genomic organization between organisms. Almost all high-C, all papers that analyzed genomic organization analyzed each organism independently, and here we want to propose a novel approach for interorganismal study of genomic organization. And we want to identify groups of gene families that underwent changes, coordinated changes between these two very distant organisms. So we begin with a model of distances between genes in S. cerevisiae and distances between genes in S. pombe. And in order to align the two graphs and compare them, we need some set of universal coordinates. So we use orthologous gene families. We used arbitrary numbers here, but the numbers correspond to the same families on both sides with the respective distances between families in S. pombe and in S. cerevisiae. Next, we want to identify pairs of gene families that are significantly closer in one organism and dispersed in the other organism. And to this end, we look at the distribution of distances in the two organisms and look for pairs of families that are, for instance, at the top region of the distance distribution in S. cerevisiae, and the bottom, the same pair is also in the bottom region of the distance distribution in S. pombe, and we denote them as interacting or co-localizing in evolution by setting an edge between these pairs of families. We can do the other way around to find S. cerevisiae families that are significantly closer in S. cerevisiae compared with S. pombe. And now we have a graph of interactions between families. So next, we are looking for modules that contain multiple families, sets of families that are significantly enriched with such interactions. And to this end, we use a probabilistic density score and look for heavy or dense subgraphs in these networks. This score is based on two hypotheses. The first hypothesis, the null hypothesis, is that the interaction that we are seeing between a pair of families could arise by chance. And we model this using RAB, which is estimated from rewired degree-preserving networks. And the alternative hypothesis is that the interactions that we see between families are indeed valid. And this is modeled by beta, which is a high number between 0 and 1. And so we have this score, this log ratio, for each pair of families. When we look at a model that contains several families, we sum over all possible pairs of families inside the model. And in order to find dense subgraphs, we try to maximize this score by trying to add more and more nodes that maximize this score iteratively until we reach an equilibrium. So we were able to identify a few dozen such models in each one of the organisms. And next, we wanted to do functional enrichment and expect to find inside these models enriched functions that are related to the adaptation of each of these organisms to their particular lifestyle. I will give these are a few examples for protein complexes that we found enriched within these co-evolving models. Some of these complexes have already been identified in the past as particularly co-regulated in one organism and not the other organism. And some of them are related to functions that have evolved differently in these organisms. Finally, my take-home message to you is that I showed you CUFS, the codon usage frequency similarity. It measures functional relatedness and the expression similarity and can be computed for any pairs of sequenced genes that we have their sequence. It is also strongly correlated with 3D genomic organization, and we were able to use it to show a very strong level of global organization in 5 eukaryotes. I showed you some level of conservation of genomic organization between organisms, which can be used to improve 3D reconstructions. And finally, I showed you a novel method for interorganismal study that identifies functionally, biologically functionally relevant co-evolving models of gene families. Taken together, all these results suggest that there is a co-evolution between function and organization. Finally, I would like to thank my supervisor, Tamir Tuller, who will be giving here a lecture tomorrow in the morning session, Ron Pinter from the Technion, my colleagues at the Tuller Lab, my funding, and finally the organizers of this conference for the opportunity to talk. Thank you.
Thanks, that was great. Very interesting stuff. I'm curious about your how you interpret some of these correlations. It seems like we know, for example, that the codon frequency impacts gene expression through the frequency of different tRNAs and so on. And so if you have, you can imagine there's that relationship, and then maybe people like yourselves have demonstrated that gene expression relates to 3D organization. But do you think that the codon frequency is somehow directly related to 3D organization, or just as a proxy through the gene expression?
That's a very good question. At the moment, we only use this as a proxy in order to measure functional and expression similarity. I'd like to point out that usually codon usage is associated with translational efficiency, like you mentioned, but there are also many examples for codon usage in other contexts. For example, transcription factors binding sites, which also makes it related to transcription, and many more other signals that we can measure using codon usage, but are not directly related to translation. And we are trying to capture everything.
So the 3D genome organization must change in time, for example, with cell cycle. How do you think that would, if cell cycle differs between two species, how will that affect comparisons of 3D genome structure?
That's a very good question. This particular dataset, these two particular datasets that we used are from asynchronized cells. So we're limited by our resolution of the data to study changes throughout the cell cycle, but it is a great question, of course. Of course, in addition, the changes between tissues in multicellular organisms are also interesting to study. But I have no definite answer. Thank you.